That would also work. Welcome, everyone, all 6 of you, and anyone joining us from home. Today we're going to talk about chapters 13.3 and 13.4, which will finish chapter 13. We did it in 2 days, or we will have done it in 2 days. Today, we are going to talk about meiosis. Is that word written up here? Yes, meiosis. Some people call it meiosis, I call it meiosis. Meiosis is like mitosis, and we'll talk about that at the end of class or at the end of the lecture. But it's very own unique thing in many ways as well, even though it's somewhat like mitosis. You'll see some similarities, but it's a more complicated concept. Now, this unit is about sex and genetics. And between those two, to make the connection between sexual reproduction and genetics, you have to address how we make sperm and egg cells, which are called gametes. Okay, we talked about a little bit of this last time, and your reading went into this, and we'll go into this tonight. So, if you have, say, skin cells, you're going to be making new skin cells every few days as the old ones die and fall off of you. And you do that through mitosis, which we already talked about. You have a cell, you want to basically make more of that same exact cell, that's mitosis. In meiosis, we're going to talk about how you go from what we would consider a normal body cell, which sometimes they use the term somatic cell, how you go from a normal body cell to a sex cell. And the big difference is that sex cells, sperm cells and egg cells, have half the number of chromosomes as all the other cells of your body. Okay, so most of the cells of your body are what we call diploid cells. Diploid, di means 2 sets of chromosomes. 1 set from your mother, 1 set came from your father. And that means most of the cells of your body have 46 chromosomes, if you have a normal human number of chromosomes. Now, when you make sperm cells or egg cells, depending on what set of gonads you have, you are going to make haploid cells. Sex cells are haploid. They have 1 set of chromosomes, or half the normal number, and 1 set of human chromosomes is 23. You have 23 unique chromosomes, and in most of your cells, they have a matching pair for a total of 46. But your sperm and egg cells need to have half the normal number. So, in your gonads, testes for males, ovaries for females, you have special diploid cells. Most of your body, including most of the cells of your gonads, are diploid cells with 46 chromosomes. And that's what we have represented up here. This would be a special kind of cell in your testes or ovaries that is called a germ cell, G-E-R-M, a germ cell. It is diploid. Now in this diagram, this is not what the chromosomes would look like in a human cell. They only have 2 chromosomes here. A blue one to represent probably like the father's chromosome that you got, and a red one to represent the chromosome that your mother gave you. So you have what are called paternal and maternal chromosomes. And in this example, this cell has 2 chromosomes as opposed to 46. If we were to try to diagram this as a human cell, it would have 23 blue ones and 23 red ones for a total of 46, but we're keeping it simple in this diagram, so we have 2. The diploid number for this cell is 2. Now, that's one chromosome, that's the other. Now, we're in interphase. What part of interphase has DNA replication? What subphase? S! S! The S phase. S probably stands for synthesis of DNA. When you have the S phase of interphase take place, you have a copy of each chromosome that is made. And they are stuck together, and as we discussed last semester, they are called sister chromatids. These sisters are the exact same chromosome. It's a copy of itself stuck together. But they have a matching chromosome that your other parent gave you, and that matching chromosome will also copy itself so that you have 2 sister chromatids for that chromosome. And that would be the case for all 46 chromosomes in this state. And that's no different than any other cell in any other situation during interphase. Any cell in interphase eventually will replicate all of its chromosomes, will synthesize a copy of all the sets of DNA known as chromosomes. So we still have 2 chromosomes technically, even though this chromosome is actually kind of 2 copies stapled together, and same here. In meiosis, and we're going to go step by 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 step, because there's basically 8 major steps in meiosis. In meiosis, we have kind of 2 halves of meiosis. We have meiosis 1 and meiosis 2. And what we are going to find is that in meiosis 1, this diploid cell will divide into 2 haploid cells. We cut the number in half by the end of meiosis 1. So these cells that come out halfway through meiosis are going to have half the number of chromosomes. Well, we started with 2 in this example, so we're going to be down to 1. This is still considered 1 chromosome, even though it's 2 sisters stuck together. And then there's another round of division where these 2 sisters will be split apart. And by the end of all the phases of meiosis, you're going to have haploid cells, each with 1 set of chromosomes. And you're going to have 4 of them. 4 haploid cells from your original 1 diploid. So, instead of taking 1 diploid cell and making 2 diploid cells, meaning 1 cell with 46 and making 2 cells, each with 46 chromosomes, instead of doing that, because that's mitosis, we are going to talk about how we will go from 1 cell with 46 chromosomes to 4 cells, each with 23. And remember, as I discussed last time, if you have a sperm cell with 1 set of chromosomes, or 23, and an egg cell with 1 set of chromosomes, or 23 total, and they fuse together in fertilization, now you have a zygote, the beginning of a human, a single cell that's back to 46. It will grow up. It will make sex cells with half the number. If it reproduces, that individual, if they reproduce, that individual would then merge their sex cell with another sex cell back to 46. So that's the pattern of reproduction. This diagram shows all of meiosis in one diagram, which is a lot of stuff. You notice it's just cluttered with all these little diagramming points. Even though it's kind of cluttered, I do want to just kind of talk through this, and then we're going to look at a kind of a magnification of this and a blow up of this and look at the 2 halves. And you can see here in this diagram, we've got meiosis 1 there, meiosis 2 here, And each time you go through one of these rounds of meiosis, and I apologize if sometimes I say mitosis by accident, it's just a habit. Each time you go through one of these rounds of meiosis, you have a prophase, metaphase, anaphase, and a telophase. Except we call them prophase, metaphase, anaphase, telophase 1, and prophase 2, metaphase 2, anaphase 2, telophase 2. And again, you can see this cell will divide, and then each of those cells in the second round of meiosis will go through a similar process and divide again, but it's not the same in the first half and the second half. So we're going to look at the first part of meiosis, look at the second part of meiosis, and then move on and wrap it up in a few slides on some details. Now, if I were you taking notes, and I would do this in the reading as well, probably, we're going to start with prophase 1. This is the first step after interphase is done. We're going to start with prophase 1, and I would, in your notes, write prophase 1. I would not write all this stuff up here, but there will be events I'm going to kind of list off, and I would just take a quick note of each one, and we will be practicing this again. And that's what your worksheet on the coursework page is dealing with as well. So prophase 1 is prophase. You know about prophase, and in some ways it's no different from prophase of mitosis. Except more is probably going to be happening in every one of these than in mitosis. What are some prophasy kinds of things that happen in cells that are going through this phase? What happens in prophase from last semester? I will not stand here in silence! Tell me anything that happened in prophase in mitosis from the end of last semester. Hey, look, it says fragments of nuclear envelope here. What does that mean? What does that mean? If you have fragments, what's happening to the nucleus? Yes! So here are some things that happen in prophase 1. The nucleus breaks down. Remember that? Y'all ate too much candy over Christmas.
So prophase 1, nucleus breaks down.
Prophase 1, the nucleus breaks down. That's something that happens in every prophase. Remember that for prophase 2. So the nucleus is going to break down. What starts forming?
Yep.
What starts forming?
Oh, the street.
The spindle fibers. Spindle fibers begin to form. Those two things happen in every prophase. Mitosis, meiosis 1, and meiosis 2. So prophase 1, the nucleus is going away, it's breaking down, but the spindle fibers are starting to kind of build up and extend out. Now, here's something that doesn't happen in any other prophase. Prophase 1 is special in this way. The homologous pairs, the matching mother-father pairs of chromosomes find each other and pair up, which is kind of a weird process, to be honest with you. So, both of your chromosome 1s will find each other. Both of your chromosome 2s will find each other and pair up together. That does not happen at any other time. Not in prophase of mitosis, not in prophase 2. This is unusual. Now, remember, these chromosomes are two sisters stuck together, and two sisters stuck together, and they pair up. Your book will use a term called a tetrad. T-E-T-R-A-D. Tetrad. Tetrad. Kind of like the game Tetris. What does tetra mean? 4. Two sisters and two sisters of each pair come together, and you kind of have this like 4 copies, 2 from one, 2 from the other, but they're matching chromosomes. They carry the same genetic information, same genes. They pair up. And when they do that, an event occurs that we're actually going to talk about at the end. The event is called crossing over. Crossing over. Like a crossover. That's in basketball, right? Anybody know? Crossover? Yeah. Cool. I don't watch much basketball. We will talk about what crossing over is because it is probably one of the major contributors to sexual reproduction being such a successful strategy for some living things. So, nucleus breaks down, spindle fibers appear, chromosomes pair up and cross over. We'll talk about what crossing over is later. All right. Prophase 1 is complete. Now we move on to metaphase 1. What's a metaphase-y kind of thing to have happen? Think back to last semester in mitosis. What happens in metaphase? The what? Yeah, the nucleus has finally completely disappeared. I wouldn't write that down. We've kind of addressed that in the prophase breaking down of the nucleus part. What's looks metaphase-y here?
Stuff is getting like condensed down.
Yeah, that's actually something, if you didn't write this, which I didn't say to, so you probably didn't, the chromosomes in prophase 1 do become shorter and thicker. That is something that always happens during prophase. Chromosomes become shorter and thicker. I forgot to mention that. But that's already happened in prophase, that the chromosomes are shorter and thicker. What's happening here? Yes! Thank you for reading that. The chromosomes line up in the middle. Okay? But there's a detail I want you to expound upon here. So, like any metaphase, chromosomes are going to line up along what we call a metaphase plate, just an equator or central axis of the cell. The chromosomes line up in the middle, but they do so in a unique way that is actually different from mitosis. Here, what we see is pairs line up side by side in the middle. Okay? Homologous pairs, which is the name for a matching pair of chromosomes, homologous pairs line up side by side in the middle of the cell. So they line up side by side. So they're side by side, the father and mother, maternal and paternal, paternal and maternal. The matching pairs line up side by side in the middle. That's only really one event in metaphase 1. I'm going to give you a second one here in a second. But that first event that you want to note for this phase is the homologous chromosomes line up in the middle side by side. The other thing that happens is that the spindle fibers attach to the chromosomes. And that always happens in metaphase. That was the case in metaphase and mitosis. In mitosis. So here in meiosis, we are at metaphase 1, and these spindle fibers are going to start to attach to these chromosomes. All right, moving on to anaphase 1. What's something that always happens during anaphase?
Separation.
the separation happens. That's right. We're pulling things apart, and that's what anaphase is about. So, before we mention separation, anaphase 1, the spindle fibers get shorter. Okay? The spindle fibers shorten. And this is critical because I ask these kinds of questions on quizzes and tests all the time so that people will miss them and I can take points away from you because it makes me feel better about myself. When these spindle fibers shorten, you have a pulling apart. What is being pulled apart though? What's being pulled apart here? Sister chromatids are not being pulled apart. The homologous pairs are pulled apart. That is almost verbatim a question that you will have. Referencing anaphase 1, the way you say it is homologous pairs are pulled apart. The sister chromatids are still stuck together, but the pairs are pulled apart. The matching pairs. So the chromosome that your mother gave you is going to go to one side, and its matching pair that your father gave you will go to the other side. Now, they don't all, like not all of your maternal chromosomes go to the same side. It's random, depending on just how they lined up. So basically, one set of chromosomes gets pulled to one side, and one set gets pulled to the other side. That's what we mean when we say homologous pairs are pulled apart. That'll probably make more sense as we get to the end of meiosis. All right, that was pretty quick. So we're at telophase 1. And of course, what do we call this groove?
Cleavage furrow.
It's a cleavage furrow. And that cleavage furrow is going to indicate cytokinesis, which we can mention in a second. But let's talk about things that happen in telophase. All telophases. Telophase of mitosis as well. Now that these chromosomes, these homologous pairs have been pulled apart, what happens in telophase? Tell me some things.
I have one question.
Okay, question quick.
Telophase 1 and cytokinesis are the same.
Cytokinesis is, and this goes back to kind of last semester, just to remind everybody. Cytokinesis is the actual splitting of the cell into two cells.
So they are combined.
Yeah, right now they're stuck together almost like one cell, but cytokinesis is the actual splitting. It is an event that happens in telophase. So you could say as the first thing under telophase 1, cytokinesis occurs, but that does typically happen towards the end. But as cytokinesis is occurring, as say in animal cells as this cleavage furrow is forming and the cells are splitting in two, what's coming back and what's going away? The nucleus comes back, and what's going to break down at this point because we don't need it anymore? The spindle fibers break down. So it's kind of the opposite of prophase. In prophase, the nucleus went away and the spindle fibers showed up. Now the nuclei, both the nucleus of both of these cells, the nuclei will reappear, but the spindle fibers break down. Now, fun fact before we move on, this is our halftime show of between meiosis 1 and meiosis 2. I believe it is at this point in females, all of this occurs before you're born. And then your egg cells go on pause until you start your cycle later in life. And at that point, every 28 days, you have meiosis 2 take place. Now in males, once you start producing sperm cells, at whatever point during puberty, then you will go through meiosis 1 and meiosis 2 like every few days and produce many, many sperm cells all the time. But it's kind of fascinating, as if you're a female, you are born with all the egg cells you will have in your life, but they are kind of stuck and on pause at this point, at the end of meiosis 1. Whoa, there we go. This is meiosis 2. Now again, between meiosis 1 and meiosis 2, you might have very little time, or you might have, 10 to 15 years of wait between these two, depending on what kind of sex cells are being made. But we now have two cells that emerged from meiosis 1, and they are going to enter meiosis 2, starting with prophase 2. And just look back to prophase 1 for some of these answers, not all of them. What are some things we expect to happen in prophase 2?
Spindle fibers here.
Spindle fibers show back up,
Nucleus.
and the nucleus is going to break down. But that's about it. Oh, and the chromosomes become shorter and thicker.
Does that happen in prophase 1 too?
That happens in prophase 1 as well.
What did you say?
About in prophase 2,
Chromosomes enlarge.
yeah, the chromosomes become shorter and thicker. They actually become longer and thinner, and you can't see them with a light microscope at the end of telophase 1. But they're going to condense back down, become shorter and thicker. The nucleus is going to go away, and the spindle fibers show up. Those are the exact things you wrote for prophase 1, and it's happening in prophase 2 again. How is this cell different, though? How is that cell different? Let me actually go back real quick. Notice we have 1, 2, 3, 4, 5, 6. 6 chromosomes. That's how many we started with in this example at the very beginning. 3 pairs or 6 total chromosomes. By the time we are starting prophase 2, how many do we have? 3. We are at half the number. This is already what we would call a haploid cell. I know you could argue, well, each one has 2 sisters, but we still count these 2 sisters stuck together as one package or one chromosome. So we've already cut the number in half. And the reason I say that is sometimes, again, for quizzes and tests, I get a little tricky. And I might say like, tell me what event this is. The nucleus breaks down and goes away. Well, you should think what word? Prophase. But then the question is, am I talking about prophase 1 or 2? And what I have to do to specify that is say, does the nucleus go away in a diploid cell? Or does the nucleus go away in a haploid cell? And haploid is what you have in meiosis 2. Meiosis 2 are going to be haploid cells throughout the entire process.
And meiosis 1 is diploid.
And meiosis 1 is diploid at least until the 2 cells have split during cytokinesis. But I try not, I say tricky, but I try not to make it intentionally, I don't try to be a trickster. I just needle at detail sometimes. But yeah, that's it for prophase 2. It's basically prophase 1, except there's no crossing over. No crossing over in prophase 2. That is a prophase 1 only thing. But your nucleus goes away, spindle fibers show up, and your chromosomes become kind of shorter and thicker again after having become long and thin at the end of meiosis 1. All right, metaphase 2. Chromosomes line up in the middle, spindle fibers attach. But here's something that's a little bit weird. By the way, and before I tell you the weird part, notice we don't have any pairs anymore. We already separated our pairs in anaphase 1. The pairs are separated. So all your chromosomes are just lined up in a single file. They're not next to anything this time. I guess you could say they're next to their sister chromatid, but they're stuck to their sister chromatid, so of course they're next to them. The chromosomes line up in the middle. But can anybody tell me how, in a weird detail, this is different? And you might think, well, it's just how they drew it, but it actually happens this way. Yeah, they line up in a perpendicular manner. So whatever way they lined up in metaphase 1, it's 90 degrees shifted in a perpendicular way in metaphase 2. Isn't that weird?
That's insane.
Yeah. What's up? But they line up and spindle fibers attach in a haploid cell. So, in anaphase 2, stop me if I'm moving too fast, but I'm almost done. In anaphase 2, the spindle fibers will shorten, which is always going to happen in anaphase, whether it's anaphase 1, anaphase 2, or an anaphase of mitosis. The spindle fibers shorten. As Ian mentioned, that causes chromosomes to pull apart. But what is pulled apart this time? The sister chromatids are what are pulled apart. Not the matching pairs, but the actual sister copies called sister chromatids. They are pulled apart from one another at this point, which is actually what happens in mitosis. This part is very mitosis-y. So your sisters are pulled apart in anaphase 1 because the spindle fibers are shortening. And then finally we have telophase 2. Telophase 2, you are going to have the nucleus reappear. The spindle fibers go away. You could write this back on telophase 1 as well. In both telophase 1 and telophase 2, the chromosomes become long and thin. They don't have that short, thick appearance that you can see under a microscope. They become long and thin and sort of seem to disappear, but they're there. And cytokinesis is going to occur. Again, in both of the cells, and the result will end up being 4 cells, and it's drawn a little funny because one of these chromosomes is so long here, but that's 1, 2, 3 chromosomes in each of the 4. We started this diagram with 1 cell that had 6. Now we're down to 4 cells that have half that number, or 3. So meiosis cuts the number in half. So we are ending meiosis with 4 cells with half the number of chromosomes that we started with.
That's awesome.
It is awesome. Thank you for saying that. By the way, and I can't remember if your book goes into this detail, and I said this in the last video too, but in males, when this happens in the germ cells, in the diploid cells of the testes, all 4 of these cells will develop a flagellum and become sperm cells. All 4. But in the ovaries, this process happens in a very uneven way. Cytokinesis happens in an uneven way. And 3 of these there'll be these tiny little cells that aren't even used in reproduction. And only 1 of these 4 will be a huge cell that actually matures into the egg cell. So it's kind of a fascinating evolutionary strategy within a species where males go for quantity, make as many sperm cells as you possibly can. We're going to use all 4 of these cells. But females are looking at quality So they put all the cytoplasm and organelles into one of the four and make this nice, big, healthy egg. That's meiosis. That's where sperm and egg cells come from. Now, I have a couple slides that I'm going to kind of go through faster than these last two have gone. This is showing crossing over. And it's an okay diagram. This is, say, one of your chromosomes, and it's actually showing two sister chromatids stuck together here, but it's really hard to see that. This is another chromosome. But in prophase 1, when you have crossing over, what that means is that these chromosomes actually make what they call this chiasmata, this kind of X shape where they loop over one another. Now this seems to be pretty random, and it may not even happen, but it is something that, if it happens, happens in prophase 1. And these chromosomes will overlap, break, and switch pieces with one another. Almost like shuffling cards in a deck. So the chromosome your mother gives you and the chromosome your father gives you, anytime you make a sperm or egg cell, there is a chance that those chromosomes that match, when they pair up together in prophase 1, will exchange pieces of themselves and make a whole unique chromosome that has never been seen before. And then that chromosome will show up in your sex cells, one of your sex cells. It's a little easier to see this concept with this diagram. This is all of meiosis from beginning to end shown, but just basically with only the anaphase parts here. You can have, and we start with two chromosomes, the blue for the chromosome from your father, and the red we'll say is the chromosome you got from your mother. They will cross over in prophase 1, and then look at this. You can have a chromosome that is going to be just completely the genetic information your father gave you. You can have a chromosome that is just completely the genetic information your mother gave you. And then you can have these weird, what we call recombinant chromosomes that are the chromosome your father gave you, but with portions of the chromosome your mother gave you stuck to them. And then those portions that are missing from the father will go over and attach to this weird, mostly maternal with bits of paternal chromosomes stuck to them. It's all about creating diversity and variation. Again, it's like shuffling a deck of cards. Now, they only show anaphase because they're just showing the separation of these two, which is what happens during anaphase. And by the end, again, your sperm or egg cells, each one can be different because for all 46 of your chromosomes, any one of those could be kind of an unchanged chromosome from one parent, an unchanged chromosome from another parent, or a shuffling of the two of both your parents' chromosomes into a new unique chromosome. And that might be the sperm cell or egg cell that is passed on to your child. And then finally, I know I've been talking for a while, this is probably a bit of a longer lecture/video. But I just want to get through this. This takes us to the end of chapter 13. This is a diagram, and your book discusses this, comparing mitosis and meiosis. Know the difference, the general difference. In mitosis, you have a diploid cell. For humans, that number would be 46. All the chromosomes line up in a single file line. And then when you finally have the one round of division, once cytokinesis occurs, you will now have two cells. each with 46 chromosomes, each diploid, which is what this 2N symbolizes. It means 2 chromosome sets. And that's mitosis from the end of last semester. But when you make sex cells, basically in metaphase, instead of all the chromosomes lining up, the pairs are side by side. So half your chromosomes are divvied up, and then you have another round where you actually have the chromatids separate, and that process ends up giving you 4 cells with half the number you had. If you didn't have this process, if you didn't cut your chromosome number in half in your sex cells, here's the problem that would arise. If you made sperm cells with 46 chromosomes and egg cells with 46 chromosomes, when they fuse together in fertilization, how many chromosomes do you have in that new cell? You would have 92. And then that person would have, engage in sexual reproduction with another person, and that 92 would be added to however many they donated. And it would take just a few generations, and you'd have every cell would have hundreds or thousands of chromosomes. And that's just too many chromosomes for a cell to function properly. So, that is the end of chapter 13. Make sure you read it. Make sure you're taking notes. We will go through chapter 14 and 15 this month and have a test at the end of this month of January. The next chapter is about genetics, and I will have some lecture, but I'm going to tone the lecture down a little bit, and we're going to have a lot of fun sheets with different kinds of genetics problems that you'll solve. Some people really like this section because it's mostly like little brain teasers that have typically fairly straightforward solutions. So we will get to that next week, but make sure you feel good about chapter 13. You can stop the video. Bye.